Hello dear students, welcome back to Zoology online class. As today we are going to talk about YUM phase. In the last lecture we have already discussed about different stages of cell cycle where we discussed about interphase in detail. So before dealing with actual stages of mitosis, let us see some early discoveries in cell division. The scientist called as Strasburger in 1875, he described mitosis in plant cells, whereas the scientist called as Walter Fleming in 1879, he described mitosis in animal cells. Again, Walter Fleming in 1882, he used the term mitosis for the first time, whereas the scientist called Farmer and Moore in 1905, he proposed the term meiosis, and the scientist called Janssen in 1909 he proposed the term chiasma. Let's see the definition of mitosis. So mitosis term is a Greek word. It is derived from two words that whereas mitos means thread, osis means stage. So you can see chromosomes as thread-like appearance in the initial stage of division. So mitosis is defined as it is a cell division occurring in eukaryotes in which replicated chromosomes are distributed to the daughter cells in such a way that they come to have exactly same number of chromosomes as the parental cell. So here what happens is, as a result of mitosis, two daughter cells are formed, which we understand after we complete the process. And these two daughter cells will have same number of chromosomes as that of the parental cell. And that is why this division, that is mitosis, is also known as equational division. Clear? Now the question is, where does mitosis occurs? So mitosis usually occurs in the somatic or body cells. Hence, it is also called as somatic cell division. Clear? But if you recall the process of spermatogenesis and oogenesis, in this process, generally we call it as gametogenesis, there are three steps, right? First is multiplication phase, second is growth phase, and third is maturation phase. In maturation phase, where meiosis takes place, right? In growth phase, as name suggests, where grows, cell growth occurs. In multiplication phase, where mitotic division occurs. So mitotic takes place in germinal cell during multiplication phase. Clear? But most of the time it takes place in somatic cells. That is why it is also called as somatic division. But we have to remember that the same division takes place in germinal cells during multiplication phase. Now, actually what happens here is, if this is a cell having 2n chromosome number, I'll write here 2n chromosome number, when this cell divides by mitosis, when this divides by mitosis, two daughter cells will be formed, and these two daughter cells will have 2n chromosome number, that is equal chromosome number of the parental type. This happens because of splitting of chromosomes, especially by the splitting of chromatids. So if you consider this as chromosome, okay, so this as chromosome, so this is the original copy, original chromatid, and this is the copied chromatid during S phase of interphase. Clear? So during mitosis what happens is, these two chromatids going to separate, and that is how the chromosome number is going to become 2n in each of the daughter cells. Clear? Now let us start with the actual stages. As I mentioned, mitosis has two stages. One is karyokinesis, that is nothing but division of nucleus, and second one is cytokinesis, nothing but division of cytoplasm. So karyokinesis has four basic phases. They are prophase, metaphase, anaphase, and telophase. Clear? So this mitosis, it is a continuous process, but for our convenience of study, we may recognize four stages in it. They are prophase, metaphase, anaphase, and telophase. Each stage is recognized by certain important changes occurring particularly in the chromosomes. Clear? Now we'll start with one by one. So now in late G2 phase, the cell has like this. It has got duplicated DNA, and it has nuclear membrane, and centrioles have already replicated in S phase. Okay, and nucleolus are present. Clear? So these chromosomes are in the form of threads, are called as chromatin fibers. So now the cell enters into M phase or division phase. So the first division is prophase. So there are so many changes that takes place in prophase. That is why we divide into early prophase and late prophase. Now let us see what happens in early prophase. Okay, here is the diagram. In early prophase, the changes which are like the nuclear membrane. Here you can see nuclear membrane. Okay, so this nuclear membrane is going to dissociate, means it is going to disappear. And here is the centriole which have split during S phase. They starts to migrate towards the other pole. Okay, okay. From these two, centriole, it start to arise small fibers. You can see here. So these are the spindle or fibers that will start to appear. Now the chromatin fiber, it starts to condense, and when they condense, we are going to see the chromosome like structure, they become thicker. Clear with this? So these are the different processes which takes place in early prophase. So nucleus is going to disappear, as I mentioned. I repeat, in early prophase, the long, thin, thread-like chromosomes become coiled or folded to become thick and short. This process is called condensation. Second point is, each chromosome is a double-stranded structure composed of two threads or chromatids. They are held together in a region called centromere. Third one, the centrioles are duplicated just before prophase starts, hence they are two pairs. One pair begin to move away from the one pole. Clear? Now the next thing is going to be late prophase. So what is going to happen in late prophase? Okay, here is a diagram. The nuclear membrane and all completely disappear here. I'll show. Okay, see, nuclear envelope, this green color one, it is completely disappear. Here we are not going to see any membrane, and the centriole, which is at one pole, and the second one has also reached the other pole. From these centriole, it start to arise fibers in the early prophase itself, right? As I mentioned there, these fibers are of two types: Some are smaller fibers and some are bigger fibers. These bigger fibers, they are actually running from, you can see here, they are actually running from one pole to other pole, right? So these are bigger fibers, and this is called as spindle fibers. Clear? So these are actually responsible for mitosis. And the smaller fibers, here you can see smaller fibers around the centriole are called as aster rays. Clear? And the aster rays along with centriole together called as aster. Okay. So, in case of an animal cell, there are two asters are formed at the poles, and now that is why mitotic division in animal cell is known as amphiastral. Whereas in plant cell, there is no centriole, that means there is no aster formation, then we call it as anastral division. Clear? Okay. Now we'll repeat the process taking place in late prophase. First one is delicate fibrils appear around each pair and each looks like a star. Hence it is called aster. As the centrioles move, these fibrils produce the spindle. Since two asters are produced, the spindle is often termed as amphiastral. The nuclear envelope breaks and disappears, and the spindle moves to the central position of the cell. And the last point is nucleolus gradually disappears, the chromosomes distributed on the spindle. So this is all about prophase. Hope you understood the point. In the next slide, we will study metaphase, anaphase, and telophase. Thank you.